All right, guys, this is a relatively short note set. I'm going to go ahead and say it is optional. If you are confident with the information that you found through the project and through what we've done so far, then you just need to refer to that for notes on this unit. If you are not, however, I strongly suggest that you take these notes. A macromolecule is a large molecule that are formed by joining smaller molecules together. They're used for various functions in living things. We cannot live without them, and we need them in large amounts. They're also known as polymers. And a polymer is made up of monomers, and monomers are small and, well, not necessarily small, but nearly identical uh, units that are linked together. So monomers are the building blocks of polymers. Um, Polymers are built through a process called dehydration synthesis. So you have a monomer, you have two monomers. One loses the hydroxide, the other loses the hydrogen, and that forms water, right? And they're linked together by a covalent bond, and they form a polymer. Carbohydrates are one of our four major macromolecules. They're made up of carbon, hydrogen, and oxygen. Their main function is to provide immediate and short-term energy storage. So the energy that you need right now and the energy that you'll need in the short term. So it is used for storage, but short-term energy storage. Other functions, it provides structural support. So for example, in plant cell walls, it's made up of cellulose, and that's a carbohydrate, right? Chitin, which is the material that makes the shells in shrimp and lobsters and those things, and it is found in bread, pasta, honey, anything sweet, anything starchy, right? Carbohydrate. This is the typical structure of carbohydrate. It has a 5-carbon sugar, um, known as a monosaccharide. So the name of the monomer is monosaccharide, and a specific example is glucose, fructose, and galactose. So these are quick sources of energy. Sugars usually end in ose, right? And we're not there yet, but the enzyme that goes for that sugar would end in ase. All right, so there you go. The polymer for carbohydrate is a polysaccharide. Different types of polysaccharides are starch, amylose, glycogen, cellulose. Okay, they can be digested by human. Those first two, glycogen and starch and amylose, can be digested by humans, but not cellulose, right? Because of the difference in structure. So even though it's made of the same monomers, look at the structure of this versus the structure of this. And you could tell of all these complex sugars, amylose is probably the one that's easiest to break apart because of the structure of it, right? Um, Glucose is stored for using immediate energy. Glycogen is stored for short-term energy, and it is found in the liver and skeletal muscles, right? So when the body needs energy between a meal or during physical activity, glycogen is broken down to glucose to give you that immediate energy. So that's what we mean by short-term energy storage, right? It's stored in the liver and stored in the skeletal muscles in the short term. But when you need it right away, it's broken down into glucose and you use that for your immediate energy. Carbs release 4 calories per milligram. The test, to test for carbs, you use the iodine test. When starch is present, iodine turns blue-black. All right. So if you have a potato, and you guys should try this at home, if you have some iodine in your home, find your iodine and get a potato or a piece of bread or a cracker, put it on it, and it should turn black or dark blue. Lipids, also known as fats, they're made up of carbon, hydrogen, and oxygen, right? Main function is to provide long-term energy storage. Other functions are providing insulation for organs, making hormones, making components of the cell membranes, which we'll talk about a little bit later. They're found in fats, oils, and waxes. Lipids, they don't have any true monomers, but if you're ever asked, fatty acids and glycerol is your answer. So it's not a typical monomer in the sense that we think of monomers. Um, Polymers are like fats and oils, phospholipids, steroid hormones, etc. The lipids store 9 calories per milligram. And do notice that that is twice as much as carbs. A saturated lipid is one that has no double bonds, right? So this is going to be there because it's always there, but you're looking for double bonds between the carbons. And as you can see, there are no double bonds between any of these carbons. It is saturated. There is no place for um, another hydrogen bond to go. So because of this, saturated lipids, they stack very neatly and store a lot easier, and that's why saturated fats are they store better. Unsaturated fats there are double bonds in the hydrocarbon tail, and they can accept more hydrogen. So here's a double bond, and this carbon can break and accept more hydrogens there. Special lipids are your phospholipids. They're responsible for the structure and function of the cell membrane. So we're going to talk about them as we go further. They have a hydrophilic head and a hydrophobic tail. So this part is polar and that'll interact with water. But this part is nonpolar and it will not interact with water. And that is important because that is what helps to form the cell membrane. So they turn in towards each other and they create a barrier to keep water out. Steroids are substances such as cholesterol and hormones. Despite their bad reputation, they are important and they're necessary, right? So there's good cholesterol, and they usually are the starting points for lipids such as vitamin D and hormones such as estrogen and testosterone. So necessary, very, very necessary. Proteins, they run your body. And we're really going to dig into the idea that proteins are very important, arguably the most important things in the body. Proteins are diverse, they're abundant, and they have many, 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 many uses. They're made of all 6 essential elements. So back in that previous PowerPoint, we talked about the elements that are necessary for life. Protein has all of them, right? Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And great sources of proteins are meat, eggs, and nuts. Proteins store 4 calories per mg. They serve a wide variety of functions. They are also the last thing your body will break down for energy, for what I just said before, because they have a whole bunch of functions. And so if you don't need to use them for energy, you don't want to do that. All right. The monomer for a protein is an amino acid. The polymer is a polypeptide. And protein has special bonds, which are called peptide bonds. Sometimes they like to give you pictures in the EOC and ask you to either identify the peptide bond, and a peptide bond always has C double bond O and a nitrogen. All right, so this is a peptide bond. Obviously, peptide bonds are only found in proteins. Some of the functions of proteins involve they act as enzymes. They control the rate of reaction for example, of an enzyme is catalase. They act as hormones. They regulate cell processes, for example, insulin, and as we mentioned before, estrogen and you know, um, they help to make bones, muscles, and skin, for example, collagen. They transport substances in the body, for example, hemoglobin, which is found in red cells, which transports oxygen, and they are a part of antibodies. So many, many functions. So if you don't have to use your proteins for energy, you don't want to. As a matter of fact, if you are to the point where you're using your proteins for energy, it means that you've depleted your carbs, you've depleted all your fats. So imagine what you look like. You're like skin and bones. That actually is considered malnutrition. That would be protein energy malnutrition if your body is using proteins for energy. The structure of protein. Protein has several different structures, and they depend on, the structure of the protein depends on its function. So a primary structure, which is just a chain of amino acid, that's the primary structure. The secondary structure is when it forms helices, right? The tertiary structure is when those helix kind of fold in on themselves, and then the quaternary structure is when you have different pieces of protein coming together to form one thing. All right, so it depends on the function of the protein. The structure depends on the function. Nucleic acids, their main function is to store and transmit genetic information, for example, DNA and RNA. Get your DNA from your parents, not your food. So even though the food has DNA, that is not the process of making DNA, and that's something we'll go in depth with later on. All right, it's made up of carbon, nitrogen, oxygen, phosphorus, and hydrogen. Should have said that second. Chomp. And it is never, ever broken down for energy. Nucleic acids, their structure involves a phosphate group, a carbon, a sugar, and a nitrogenous base, right? So all three of these things join together covalently to make a nucleotide, and a bunch of nucleotides join together to make a nucleic acid, right? The polymer, I just said it, is called a nucleic acid, and there are two examples, DNA and RNA. All right, that's it, guys. Make sure that you draw the pictures. Again, this is a great resource, but everything that's contained within this PowerPoint, you should have researched and uncovered for yourself in your project. See you in class. Come with questions.